So that was looking at a single time point, single data set using our default painting configuration that uses the full color range and fits it to your data, and just painting onto the overview. Now, one thing I should emphasize right here is that if you use the default coloration, from experiment to experiment, the color where the colors are placed will change, because, for example, this particular column happens to max out at a full difference of 3.4 log units. If another one maxes out at a full difference of 6 log units or of 2 log units, that will also be colored red, and the color scale will be extended or compressed appropriately. And this means that you cannot, just off the top of your head, throw things at the default color scheme and compare them. But there are a couple ways we can compare them. We can set the color ranges ourselves, we can set color cutoffs, or we can do an animation. So I'm going to show you all three of those options. Okay, here we are back at the page. Before I show you those options, let's take a quick look at one of the other display options, which was to generate a table of individual pathways exceeding a certain threshold. So we're going to click on that, and let's set our threshold here to 0.6. So we're looking at log units here, and a 0.6 threshold is about 4-fold. And I'll hit submit. Okay, here we are with the table that's been generated by setting this 4-fold cutoff. Now the table has kind of large cells, and let me show you the culprit for that. It's the tRNA charging pathway. So this is actually a bunch of different tRNA charging reactions that are all stuck together in a kind of ad hoc pathway. So it's stretched out the page here, but there's some really interesting stuff here. So here, you should be probably familiar with this. We have this pathway here. We'll just scroll down. You'll see other pathways. You'll notice that again we're showing you all the isozymes, all the different genes that are involved. Scroll down, and you just get a list of pathways of interest. So if you don't want to look at the overall display, if you really do want, any pathway with a gene whose expression has changed, in this case 4-fold or more, then you can do something like this. You can set your own cutoff, get a pathway list, get everything painted onto the pathways, and have another convenient at-a-glance intuitive view of just what your omics results mean. So now we'll go back to the query page once more. And now let's play with the displays a little bit. So, for example, let's go back and click on Paint Data on the overview chart. And you'll notice that I've been able to go back and do things over and over again without having to tell it to reset the different characteristics that we're looking at. And this is handy, because if I scroll up the page here, you'll see I'm still on the same data column. I'm still looking at E. coli K12. I still have the same file I've uploaded, and it's just going to assume I want to do everything the same way until I tell it different. So hey, let's tell it a little different. We'll switch up to column 7. Now going down here, let's go to a full color spectrum with a maximum cutoff. So a maximum cutoff, we'll do 0.6 again. A maximum cutoff basically sets the outer ranges of colors that you're looking at. It sets the red and the yellow to the value I'm putting in here. And of course, I'm going to end up with a lot of stuff above and below that range just living in red and yellow. So this is one way to say, well, I really just care about everything above or below an n-fold change. In this case, again, a 4-fold change. So I'm going to do this and leave it at Paint Data and hit Submit. Okay, so here we are looking at another single data set having capped our display colors. And so you'll notice we still have the full color range, but now you're probably seeing a lot more red and yellow because we've set those as our certain cutoffs, our 4-fold cutoffs. So down here we have expression ratio chart. We can see that the histogram is a lot fatter. It's kind of squished because the upper bounds dropped by quite a bit. So I'll move over here, and you can see this actually ends up being a sort of more colorful display because we've included a lot more of the bright outside colors by setting these cutoffs. And basically, this is one way to, at a glance, get things above and below a certain cutoff while still seeing what the gradations are in the middle. So that's one way to do it. And this is one way that you can set your data sets to be all comparable with each other. You may not want to set the cutoff as low as I did. If you're looking at something where you know that you get a 3.5 log unit swing in at least one of the values, maybe you set all of your cutoffs to 3.5 log units so you can compare. And you'll see that the software does that automatically when we do an animation, which I'll show you in a bit. But now let's go back and look at one other way that we can set the colors, and it's a way that is my personal preference. Okay, so we've done a full color spectrum computed from the data provided. We've done a full color spectrum with a maximum cutoff. Let's do 3-color spectrum with a specified threshold. And I'll do that. And now watch what happens. Okay, so here we are. We're once again looking at a single column, single data point. We're looking at it on the Omics viewer, and we've set a 4-fold cutoff above and below, and basically now we have a 3-color display. And so I like this display a lot because it has that kind of intuitive, I'm going to see exactly what I care about kind of look. So anything that's red is above the 4-fold cutoff. Anything that's yellow has gone down 4-fold or more. And remember, the 4-fold is arbitrary. I set it myself. You can set whatever you want. And blue occupies the entire middle range. And so we get this kind of display. And now I'll just click over to the right and show you our histogram, now completely clunky, but it shows you where things live in the display. Click back over on the left. And again, if you go over one of these pathways, you're going to get the pathway display. You're going to get the stuff painted on it in the 3 colors. And you get isozymes painted in different colors when present. But you notice, since I've collapsed the color ranges so much, we no longer have the gradations. A lot of the isozymes fall into the same brackets. And since they fall into the same brackets, they're going to be painted the same color. There's no reason to have multiple lines. So now I'll close that. Okay, so now we've done a quick overview of a couple different ways you can look at a single data set on the Omics viewer. So now, with that in mind, let's take a look at how we can look at a multiple data point set. And the typical example here is going to be a time course.